I think we can go ahead and get started. So thank you again for joining us today. A reminder that the public webinar series is designed to educate attendees on the scientific ecosystem, and every month we'll have a representation from a data coordinating center and new developments from the centers that support them. Today, we're going to share some scientific updates from the Knowledge Center, upcoming events within the CFDE as a whole, and then you'll hear from our featured speakers. Just a reminder that if you are unable to attend any of the previous webinars, they are all available on our YouTube page, which is linked here, and you can go through any of those videos as they're available to you as needed. As the KC, we love to create use cases around how you, the researcher, use data sets within the DCC. This is our homepage for the KC. If you have any feedback for us, any questions, or would like to be added to our listserv, please email us at help@cfdeknowledge.org. So for some events that are coming up within the year, we have the ASHG, or the American Society of Human Genetics conference in November. This is an even larger conference that is involved in genetics and genomics. So our exhibition booth will feature DCCs, including GTEx, hopefully Kids First, and a few others. The two centers, so the KC, the DRC, and hopefully the ICC as well, to go over the CFDE as a whole. So we're really looking forward to your participation. If you're interested in representing CFDE, please shoot me an email or reach out to our help email. The more people that are attending, we'd really love to have that be available. In addition to the booth, we'll also be hosting an ancillary session. So this will be Wednesday, November 6th, 11:45 a.m. to 1:15 in the convention center. We'll have some more details on the ancillary session as we get a little bit closer to the date. So please keep those on your calendar if you'll be attending ASHG. Some other events that are within the ecosystem that we'd like to socialize include the CFDE virtual workshop hosted by xRNA, the DRC, and GTEx on September 12th. If you plan to attend this event, the register to deadline is the September 10th. And HubMap is hosting their fall 2024 hackathon, which will begin on September 20th and wraps up on October 4th. Today, I'd like to introduce our speakers from Acute to Chronic Pain Signatures, an initiative to identify biomarkers and advance pain science. A2CPS is a new initiative within the CFDE. So I know I've showed this slide almost every webinar series. They're upcoming, so they'll be added to this as we go along. So we'd like to welcome them to the CFDE today. And just quickly, the program is geared towards identifying biomarkers and advancing pain science. So with that, I will turn it over to our speakers from A2CPS.
Hey, let me see here. Let me see if I can share here. Hi, my name is Martin Lindquist. I'm the PI for the Data Integration and Resource Center for A2CPS, and I'm joined by Ari, who's in charge of the DCC, and Seth, who's a new researcher with the consortium, who's worked a little bit with Common Fund data. So I'm going to start out by just sort of presenting A2CPS and what type of data we have. Ari will continue to talk about some of the sort of data formats and accessing A2CPS data, and then Seth will talk about some of his experiences with sort of playing around with CFD tools and using CFD data to answer interesting questions. So with that, I'll just get started here. So chronic pain is a huge problem in the US, and it's actually very, very poorly understood. And so the goal of our acute to chronic pain signatures program is to sort of understand the transition from acute to chronic pain. And so it turns out that, roughly between 20 to 50% of people will transition to chronic pain after a surgery, or where the surgery is the acute pain episode. And the sort of the mechanisms driving these transitions are very poorly understood. But it's sort of hypothesized that biological, psychosocial, and social factors have a huge impact on driving this transition. And so the goals of A2CPS are as follows: is to identify biomarkers that can predict both the susceptibility but also the resilience to the development of chronic pain after an acute pain episode. We have three aims, and these are sort of provided to us, through the RFA. But what we want to do is to begin by using a sort of a candidate approach to determine whether certain predefined biomarkers across sort of multiple domains are individually predictive of the development of chronic pain. We also want to combine these into so-called biosignatures, which are sort of combinations of biomarkers, to determine whether they can improve the prediction from acute to chronic pain. And then finally, we're going to have a very novel and exciting data set, and we're going to do sort of exploratory and discovery validation to define new biomarkers and biosignatures that predict the chronic pain. And so the participants that are in our study, so A2CPS will ultimately consist of nearly 3,000 participants, so 2,800 participants currently, from two different acute pain cohorts. The first consists of individuals that are scheduled for a total knee replacement. And the second is participants that are scheduled for thoracic surgery, and that's often participants that have a cancer or something that should be removed from sort of the chest area. We started recruitment over 3 years ago, so in March 2021, and we were slightly delayed by COVID, as a lot of studies were. And we've currently enrolled about 1,600 participants of our goal of 2,800. And enrollment is sort of rolling on quite nicely now, and will end next November, after which time we'll just acquire data on the participants that we have enrolled by that time. What type of data are we acquiring? But it's a very wide array of data. Some is sort of psychosocial and behavioral data, which is obtained via patient-reported outcomes. So participants will answer questions about sort of depression and childhood adversity, catastrophizing, different types of things about their sleep and cognitive functioning, etc. We also have a lot of omics data. So we have genomics, transcriptomics, proteomics, metabolomics, and lipidomics. We have a battery of neuroimaging measures. This includes both structural MRI, diffusion-weighted imaging, which can tell you about the fiber tracts, and as well as a couple of functional MRI runs that can tell us about sort of connectivity between different regions. And those connectivities both acquired during rest and while the patients are experiencing a painful stimuli. We also have quantitative sensory testing, which are a few things to assess people's pain levels, and we have functional testing, which is basically whether you can get up and down 5 times, whether you can walk 10 meters, and for the thoracic patients, is about whether their breathing is impaired. And finally, we're going to have an electronic health records pool. So this is a very wide battery of different data types. We're very excited about, and we're collecting on these 2,800 participants. The outcome that we want to predict at the end of the day is 6 months after the fact. We're going to do a sort of a 2-week trajectory until we get 7 days worth of data. And basically the worst pain they've been feeling the last 24-hour measured over a 7-day period. And typically, if you still have sort of pain in the areas of surgery 6 months after the fact, then it's deemed to be chronic pain. We also have a bunch of secondary and exploratory outcomes, which is related to disability, opioid use, and misuse, and pain interference, etc. Our primary biomarkers, again, we have roughly 40 of them, 36 at this point. They were selected on sort of previous literature, and they sort of are roughly 15 from the psychosocial measurements, 10 from the omics, 8 from the brain imaging, and 3 from the QST measures. So together that we're going to look and see how they individually predict chronic pain and the other measures, but also how they together interact and predict it. This is our data collection, and so we have data is collected sort of longitudinally, and so basically what we have is prior to surgery, which is we have people answer 30 or 40 different PROs, or patient-reported outcomes. We bring them in on site, and we measure QST, omics, take a blood draw, physical function, and imaging. So this is what we call our baseline measurements, and those baseline measurements are what goes into our biomarkers, because we want to be able to predict chronic pain at a later date based on these prior to surgery. So we want to be able to predict who's going to be susceptible for chronic pain and who's not. After surgery, participants get a daily text asking how they're doing, and so we have this daily items for a month after surgery. After 6 weeks, we bring them all in again and take a second blood draw and get all the omics again. And at 3 months, we repeat the whole baseline experiment again. So finally, at 6 months, we measure our primary, secondary, and exploratory outcomes, i.e., if they're still feeling pain, if they're using or misusing opioids, etc. At 12 months, we also have a voluntary survey, but a lot of people have been filling that out. So in general, for the main goals of the study, we calculate biomarkers based on the baseline data, and we try to use it to predict the 6 months outcomes. We also have a great opportunity to understand the mechanism of this through the sort of longitudinal study. So we have three omics time points and 2 imaging time points. So I think we'll have a rich data set to explore sort of differences in both these multi-omics and imaging in who sort of is resilient and who's susceptible to chronic pain, which is really exciting. This is, of course, a lot of people involved with this study, and so we have 2 multi-site clinical centers, and they consist of 6 clinical and 13 recruitment sites. That's a little that number should be upgraded. It's probably closer to 18 recruitment sites now. We have 3 omics data generating centers that we work with to get. So UC San Diego processes the genetic variants and xRNA, PNNL, and together with Pittsburgh, do proteomics and Luminex, and Wake Forest and UC San Diego, UC Davis, sorry, do the lipidomics and metabolomics. We have a clinical coordinating center who takes care of the joint IRB, and then we have the data integration and resource center, which is where we're from. And so we coordinate data collection, transfer, storage, track where everything is, and also analyze the data and do outreach to the public and scientific community. So that's our task, our charge. Well, recruitment, again, as I mentioned before, is expected to end in November 2025. It'll take roughly a year and a half to sort of close out the project to get all the data up on its final resting spot, which is the NIMH Data Archives, NDA. Before that time, though, we are going to make baseline data consistently available. We can't share post-baseline data until the end of the study by rules of the consortium, but all baseline data will be made available. And the first external release is planned for this fall. So that's really exciting. So that first release consists of the first 809 participants. And these are people that had surgery on or before the last day of February 2023. And basically, these are all our data. The colored ones are all data that's available. The gray is no such data. But in the first release, we're making sort of demographics, psychosocial variable, QSD functional testing, and the imaging available. In a second release that's coming pretty soon afterwards, we'll also make the EHR and the omics available, as well as the computed biomarkers. And then from there on, we'll have continuous, we'll have periodic releases of everything in this sort of first row. The timeline for that is as follows. So we usually have an internal release, 6 months before the external release, where we sort of can work out bugs and look over some of the things. And then, we have an external. So our first external release is scheduled for October, November of this year. And it's going to be followed by, adding the EHR and omics data in a few months later. And then, we from there on, we will have sort of yearly releases with increasing number of subjects. So release 2.0, for example, that will come in the late spring of next year will consist of 1,400 participants. So we're sort of building up from 800 to 1,400. Maybe this release will be 2,200, and then finally we'll have 2,800 people. What are some exciting and unique aspects of A2CPS, which I think could be pretty interesting for the folks here? One of the things is again, we're a Common Fund project, and we've been working with the Common Fund data ecosystem, but we're also kind of related to the HEAL initiative, which is the big to stop the opioid epidemic and to help end addiction in our lifetime. And they also have a data ecosystem. So we've been working actually on harmonizing both with the Common Fund and also with HEAL as much as possible. So I think we're in an interesting position where we can sort of serve as a bridge between Common Fund projects and HEAL projects, which are of course two very big sets of studies, and so that's an interesting thing. A2CPS has already collected imaging data on close to 1,300 people with at least one measurements, and 800 of those are with two measurements. That actually makes us probably would make us a top 10 project in the world among sort of sponsored neuroimaging studies. The biggest is, of course, the UK Biobank, which is close to 100,000. That's hard to beat, but A2CPS is already bigger than the Human Connectome Project young adult cohort, which was for a long time sort of the seminal project, the benchmark project for large neuroimaging studies. So it's exciting, but I think that there's some things that we have that are sort of unique. We have this big neuroimaging cohort, but we also have this sort of longitudinal multi-omics data set. So if one can find sort of interesting sort of omics markers, and we can study them in A2CPS, and then we'll have brain imaging data on those particular participants. So I think there's a lot of sort of unique things about A2CPS that is sort of intriguing, and I think that and it's excited to work with you guys to make harmonize these things. And so again, these are sort of just the acknowledgements, but before I hand off to Ari, any questions or? All right, if not, Ari, it's all you.
There's one
Oh, there
question
is. Okay. Oh,
actually. Yeah.
sorry. Okay,
No, it's
my
totally
bad.
okay. Fascinating data sets. What is the data sharing plan? Is there going to be a DRS server for access in the cloud?
Ari, I think you have to unmute yourself.
Yeah, I'm going to talk about the data sharing plan.
Awesome. Great.
Yep.
Well, thank you.
All right. So why don't you take it away?
All right. Let me see. I think this is it. All right, there we go.
You can see your screen now, Ari. Yep.
All right,
Looks great. Thank you.
thank you. So my name is Ari Khan. I'm with the Texas Advanced Computing Center. I'm part of the Data Integration and Resource Center of A2CPS. I'm also the PI for the Data Coordinating Center. And I want to tell you a little bit about how the data flows within A2CPS. So up here in the top left, we have the clinical sites. And what they're doing, all those kind of data that Martin was talking about, it's entered into REDCap, whether it's clinical data, subjects, new subjects, if they're creating or doing blood draws and then shipping those off, if brain images are being collected, the psychosocial data, the PROs, those are all collected in REDCap. And and that's connected to our virtual biospecimen repository at the DCC, and I'll talk a little bit more about that. But when the biospecimens are collected, they're after they're collected, they're shipped off to a sample management group at University of California, San Diego, and they collect all the samples until there's enough to actually send off to the omics centers for analysis. And so once there are enough, they're shipped off to the omics centers, and we provide them with run lists, basically to optimize, to minimize the batch effects that might happen, but also to optimize their assay flow, right? So we want to I guess minimize their expenses. And after they do their assays, they submit their results to the DCC here. And then the Data Integration Resource Center, the DIRC, we have access to that, and so that we can start analyzing it. So the virtual biospecimen repository, we're trying to figure out and keep track of all these samples because they're valuable. And so you want to know, like, what container they're in, which tube they're in, what box are they stored in, but where are they being shipped? Did they actually arrive at the destination? Who has them? And were there any exceptions? And is there anything left from the assay? Like I said, we also provide them run lists, and then when they submit, provide the ODGC's run lists, and then when the ODGCs submit data, or the clinical sites or the medical sites submit brain images, then we can link that data to the subjects and biospecimens that they're associated with. So we have this cool API. If we provide these slides, you can click on it and check it out. So this is how we process all the data from REDCap. So we actually have, and anytime there's some event, we get a kind of a push webhook. And so depending on whatever the instrument is or whatever the form is, we have something to handle it. And the goal is to collect all that, to collect all that data into our database. And we're using Postgres with a API on top of it. Also, whenever there's a new event or new shipment of biospecimens, we get a notification so that we can track it. So we can keep know what's going on. So our goals are to validate the data and then to link the data with all the other patient-based data. So the subjects, samples, the AHR data, psychosocial, MRIs, the omics data. And then as data is submitted, as the omic data or the imaging data is submitted, this is kind of showing a EHR centric one, but we have a data type specific handler. So whatever the data is that's being submitted, there's a specific handler. We know that a new file has been submitted, and we know what kind of file it is, and so we know how to handle it and how to validate it. And then the validated files are deposited, into directories, but we keep track of all that in our database. So data sharing. So due to the study of the design, we're not sharing anything except for the baseline data until the study is completed. So 6 months after each internal release, we're going to deposit the baseline data or the pre-surgery data into the NIMH data archive, or the NDA. So as Martin said, the first deposit is expected this fall. And then 1 to 2 months after each NDA release, right, we're also going to submit the metadata to the CFDE so that you can actually use the CFDE portal to find those data and relate them. And then 6 months after the study is completed, all the remaining data is submitted to the NDA, and then that includes the rest, the whole of all the data that we've collected. The metadata for those submissions to the NDA is also submitted to HEAL, and also to that, I didn't put CFDE here, but there too. And then if there are any leftover biospecimens, there's actually a HEAL biospecimen repository. And so we'll collect all those biospecimens and ship them over there. Okay, all right. So these are all the major data types and where they are being deposited. Basically, everything gets deposited into the NDA. Except for the project metadata, which gets submitted to CFDE and HEAL, and the leftover biospecimens. This is what this is how you get to the NDA. It's I think the NDA, a lot of the data that's or a lot of the data structures and the kind of the shape of the data is kind of being developed as the NDA progresses and as those new data get submitted. But it's a highly curated data set, which means that the metadata surrounding it has very specific data structures that we have to submit to. So this link here will go to all the harmonization standards that they have, but we can go into some of it if there's time. But basically, there is controlled access and open access data available there. One interesting aspect of the NDA is that they have these computed subject GUIDs. And the idea is that you have this base set of metadata about a subject that you put into a program, and it spits out a GUID that's specifically associated with that metadata. So it kind of obfuscates the metadata, but it provides a consistent ID so that you can find the same if that subject is part of many different studies, that you'll find that same GUID for that same subject. That makes sense. One thing that's important to know is that the NDA is on Amazon Web Services. So the download to non AWS is limited, but if you have an AWS instance of any kind, then it's unlimited data transfer. And so this is kind of what Martin was talking about in terms of like how the data kind of hangs off of time points. So you either have, you know, one of two cohorts, the total knee arthroplasty or knee replacement or the thoracic cavity, and then you have a patient or a subject, and then you have EHR data about that, and then you have time points. So the time points are either baseline or these other ones, 6 weeks, 3 months, 6 months after surgery. And all the data kind of, you know, hangs off of there. And these are the data that you'll have access to until the end of the study. How much time do we have?
About half an hour left, Ari. We have a
Okay.
question. Is now an okay time to pause for questions since we paused?
Yeah, let's do that.
Sure. So the question was from online. It's for older adults who often go post-acute rehabilitation locations outside the hospital EHR and who undergo interventions and take medications that could affect pain. Does A2CPS acquire these important data points?
I can answer that question. Yes, we do. At each time point, we ask about sort of different treatments, you know, that's been done since the last visit, and so you know, and that includes both medication and you know, seeing other professionals. It could be sort of like acupuncture or whatnot. Yeah. So yes, we do collect that.
Beautiful. Thanks, Martin. All right, Ari, how you doing? We have staff back to you. I don't have too many other things, so I think you're good to go for a few more minutes.
Okay, well, I'm, you know, I don't know if we want to go into it, but I do have slides kind of like on each data type, but you know, I guess I can go through it real quick. The clinical and psychosocial data, so there is some controlled access data, but we're basically anonymizing all of it. And so there'll be open access versions available. Again, it goes to the NDA, and the metadata standards are the NIH HEAL common data elements. And then we have EHR data, which Seth is going to talk about. They can be open access, and we link all the data to that's associated with a subject to the EHR data. The CDEs for the EHR data are going to be in the NDA. With imaging data, so it turns out that like a full image, you can reconstruct a face, and also the DICOM files have PHR, PII data embedded in them. So what we do is we create these skull stripped nifty files, and those can be open access. So those are submitted to the NDA, but we might submit them to other repositories as well. And we use the ABC metadata standards. For gene variants, those are controlled access. They're basically a SNP chip. And so the file types are IDAT and VCF. XRNA, those are open access. And if you're familiar with XRNA, you have FASTQ files and counts. For proteomics, we actually have two groups that are doing proteomics. One of them is a Luminex-based platform, and it's an immunoassay. And this is a primary biomarker. There's also a Thermo Fisher platform, and it's a spectroscopy. And so the file types are, they have a raw file or an MZ file, and then there's these other processed outputs. Metabolomics and lipidomics, they don't contain any protected data, and you kind of have these set of data that are specific to those kinds of platforms. And then there's the project metadata that I talked about that are being submitted to the CFDE and HEAL. And, it's using the, for the CFDE, we actually based our database off the C2M2 model that the CFDE uses. And so if we can submit it, when we're ready, we can just submit it because it's already ready. And then the metadata standard for HEAL is the HEAL CDEs. And that is all. Seth, wait, are there any questions?
That was great. Ari, when should users from the external perspective start to see some of these data rolling off from you as being accessible to the community? Or did you already cover that?
Great question. So they'll be, they should be available at the beginning of 2025.
Okay. Cool.
I think it's fair to set your expectations because they'll be submitted in the fall, but then there's like Thanksgiving and Christmas, So I think that's probably.
Beautiful.
All right.
Awesome. Thank you, Ari. That was great. All right, so this is actually a great setup to our next speaker, and I want to give a little interlude here to Seth because I think it's a kind of a cool bridge to the next presentation. When having heard from Ari and Martin about A2CPS, about this DCC, it's a new one, and what you've already heard is they're going to be generating a lot of data in the multi-omic space, including genetics and genomics, transcriptomics, and making those accessible around molecular signatures of pain. And that could be, relevant to so many other common diseases beyond what the primary reason that they're taking these samples from people with knee and surgery, right? So it's more useful broadly for common disease. And so as a human geneticist, I think about the utility for A2CPS broader in terms of thinking about how people's response to pain and other common diseases and how we can marry this data. But the reason Seth is here is because it's an interesting serendipity because about, a month and a half ago, Mackenzie and I attended an NIH workshop about the Common Fund outreach and how we as folks who are funded by the Common Fund or the NIH think about how to raise awareness for Common Fund resources. And Seth gave a very interesting talk about he, as a student, was trying to access data from one of the Common Fund resources, Gabriella Miller Kids First. And that's one of our resources that's very heavily genetics focused. And it's an incredibly good resource for those of you who used it for rare disease. And you can go through a process to access data. And once you access data, there's a lot of things you can do. But he described his experience trying to access data and navigate Kids First, which I thought was very interesting because it taught me some things about how that's got done, but also is really useful for the community in terms of how one would go about doing it. So Seth is kind of like a target user from a computational perspective. And what's even cooler, moreover, is he's now just recently joined A2CPS to analyze some of the data that Ari and Martin just talked about. And so this is an example of someone who actually was an outsider using some CFDA data, coming to work on another DCC, but thinking about how to integrate data and make results accessible. So I thought it'd be really cool to have him come and talk about his experiences, sort of navigating a Common Fund resource, but also transitioning that to A2CPS. Because as a knowledge center, we think about how do we raise awareness to folks like you who are listening on this call to use the resources in the Common Fund to address scientific questions. And you all have different user experiences and profiles. So Seth is just one example of a type of user. So he's going to talk about his experiences, and I thought that'd be really useful for you all. So hopefully that was a decent introduction, Seth, and I'll turn it over to you.
Yeah, thank you so much for inviting our team. And also, I was looking at the list of participants. I want to say hello to everyone I've met throughout my time at the CFDE who's watching, and to those who I have not yet met. But I'll share screen here. Let's just make sure this works. And yeah, so as discussed, as an undergrad, I was working with GMKF data on the Common Fund and initially struggled in how to navigate that data, but also how to create analytic workflows, custom pipelines to assess that data for what we were trying to find was sex-specific genomic differences underlying oral facial cleft etiology. So today I'm just going to talk a little bit about that process of creating custom pipelines, accessing Common Fund resources and running them through a pipeline to answer any sort of scientific question you may have as a user or someone who's just looking at the Common Fund utility and how that whole process works. So in general, is your CFDE analyses. And I'm going to do so through the avenue of talking about A2CPS hypothetical use case on the cloud. So yes, my name is Seth Burke. I'm a post-bac researcher. And so, right, it would start with you, the user, as a scientist who, has this whole world of trying to figure out new hypotheses, scientific investigations. And now at your disposal, you have CFDE data coordinating centers that have a wide range, as we discussed today, of data resources, data types that could be cross-analyzed across different data coordinating centers. Just to name a few, I know there's one group that looks at exercise biology, another group that does nutrition, our group does chronic pain, some with extracellular RNA, and the list goes on. And I know the Knowledge Center has a website that outlines kind of each data type and how they could be used together. But anyways, say in our hypothetical study, we're using A2PCS or A2CPS data as discussed. And as the user, you're looking at these data, and you're thinking to yourself, what kind of analysis could be run? And so say based on your background, what you know about biology and physiology, you come up with this question. Again, this is purely hypothetical, but is there a relationship between prefrontal cortex activation and C-reactive protein levels for individuals who do develop chronic pain post-surgery, right? As the user, you understand that the fMRI data could show you something about prefrontal cortex activation, and proteomics and the assays that we're running have open release data about C-reactive proteins. So with that knowledge, you could formulate a hypothesis like this, and then you have to start thinking to yourself, well, how could I now process these data from the Common Fund? And the strategy that our team used when we were working with GMKF data that could be applied to any DCC or provider was this. So we would start on our local computer thinking about the processing steps that we needed to run these data. So in our case, there's fMRI data, proteomic data. Now there could be different scripts harnessing different computational environments to process them. And what do I mean by computational environment? Well, that could be the programming language that is used to run these data sets alongside the scientific packages that map directly to the environments you need to run these data. So for instance, say we're using R and our programming language to run some sort of statistical analysis to figure out this relationship. And there's going to be packages necessary to run these data types. So for instance, there might be an fMRI software package that could process the prefrontal cortex data. And there could be a proteomic package to process the proteomics data. This is similar to what we did with the genomics data from GMKF, where we used a scientific package called TRIO to assess case-parent proband trios to find some sort of linkage between a causal variant and our outcome variable. So now that we have these packages in mind, we have to figure out a way to put these onto the cloud so we could make our analysis function on the cloud. And luckily, there have been software containment platforms that have risen in the last decade, namely Docker and Singularity. But say we use Docker. This is a very common software containment platform. And what that means is these packages that we discussed can now be put into these boxes, these computational boxes that could be put to the cloud. So then you could run these data in the cloud. So say we construct our box that has our software containment in them. We choose a biomedical cloud platform that is available to the CFDE. I worked heavily with Cavatica, but there are others under the Seven Bridges domain, and also one called Terra, which our friends here at the Broad work with regularly. And so say we choose Cavatica, for instance. Now, that Docker container that had our software that could be used to process our steps for our data types can be ported to Cavatica. They have a Docker repository service that is directly integrated with Docker itself, as does many other biomedical cloud providers that are available to the CFDE. And so now that you have your scripts and your Docker container, the next step in an overall analysis like this could be, or would be, to port these software containers onto the cloud, as discussed. Let me just check the time. Okay, we're good. And so after that, now that they're sitting on the cloud, you could create these processing steps. On Kvatica, they're called tools. On other platforms, they're referred to by different platform-specific terminology. But essentially, these processing steps are going to ultimately be used to run these data to answer your scientific question, and they contain the software packages now necessary to run these environments on the cloud, right? So you have one step for your fMRI neuroimaging data that harnesses a certain script that you created locally that has software that will now operate on the cloud. Same thing with proteomics data. And say you have another script that merges these data together to figure out some sort of linkage. So how do you connect these steps? That's the third fundamental that I'll discuss today. And the way to do that is with these custom workflows, which derive from workflow language systems, which are essentially their own programming languages that connect diverse steps that could harness different software environments together in a way where the data just flows through each step seamlessly. So the files that could be output as intermediate files don't even necessarily have to be added onto your storage, because these data that start in the cloud just filter all the way through to the end. And so in Kvatica, the workflow language is called Common Workflow Language, but there are others, namely WDL, WDL, which Terra uses, and a few more. So with these steps in mind, you can imagine a world where now you're harnessing these data from A2CPS. That's part of the NDA. You work with our team and a cloud service provider to input these data onto their service, run them through a pipeline that you customly created based on your question of interest, and then output these results for some sort of scientific finding. And maybe you download them locally so you could work on data visualization software that's native to your computer. And all for the end goal of scientific communication, right, to better the world. So once this whole process is understood, the idea is it's actually data coordinating center agnostic, language agnostic, and this way of thinking can apply to any sort of analysis that requires a custom workflow, because you have granular control over the scripts that you're creating, the software that you're running, and the cloud that you're harnessing. So you can imagine a situation where now you're choosing different data types and different data sets from across coordinating centers, data coordinating centers, putting them into your biomedical cloud platform and running any sort of investigation that's interesting to you. So as a user, that's what I think is exciting about CFDE and kind of how these pipelines can be created to answer novel science. And that's something that I hope helped a little bit out today in discussing. So yeah, thank you guys. Say thank you.
Excellent. Thank you, Seth. Can you go back to the slide for a second? I have a question for you.
Oh yeah.
I really liked how you laid this out. I think it sort of describes it from the sense that like anybody could do this sort of thing. But I'm thinking as the knowledge center, just from my own, like when you say Kvatica and results, what you'd actually also love is anybody who does this could make their results accessible on the knowledge center or through the DRC, depending on the format by which they're in, right? Because some of this you'd want to make open access to people. Is that sort of one extended use case for this in your mind?
I think so. I think any study that harnesses data that is not controlled access, such as genomics data, could be put onto a knowledge center portal as a use case that could be extendable to others interested. I agree. Yeah.
That's great. And do you imagine that for A2CPS, this is something that you hope to do? Like this is sort of the process you hope to employ for A2C for some of the analyses?
Down the line, yeah, I would hope so. As I look into PhD programs as well, it'd be something cool to do. So yeah.
Other questions for Seth, Ari, Martin? See one popping in. All right, here's another one.
Yeah.
Knowledge portals often to provide query and visualizations that do not require some substantial programming skills. Will a user interface be developed? Oh, wow. Someone was thinking like me. I love whoever you are. That's a great question. Seth, do you want to answer it from your perspective? And I can answer it. I think that's along the lines of the question I just asked, right? Which is, once that's done, you'd want it to be almost like button clickable, where you click on make this accessible via a portal. And it almost could be not a portal, but like a table. It's not a full portal in many cases. But you can imagine that could be something that where immediately we work with the DRC and the KC to either make it accessible via one of like the DRC's playbook modules. That's one option I can imagine, or through a visualization or a table that we make readily accessible on the KC as part of one of the workflows that we're building, or within one embedded in one of the DCCs if it's primarily from a DCC. I would love that opportunity. So if we could do this as a center, in fact, I think, you know, there's about to be another center awarded from the CFDE for a cloud computing workspace. Hopefully that center will help work with us on that. And that's what I'm betting will happen. Other questions? Is someone in the chat? Anything else?
So Avi just messaged, do you think you can
Yeah,
convert your
actually, he
workflow?
just-
Yeah,
Say it again, Mackenzie. Sorry, I would talk over you.
no, it's okay. Do you think you can convert your workflow into a workflow supported by the CFDE Playbook project?
I'm guessing this question is broad for me here. I would have to look in more depth with the CFD Playbook project. I know it's extremely useful and lowers the entry point for any researcher to go in and craft their own custom analysis in a different way than harnessing biomedical cloud platforms. So it's something that I'd have to look into, and I could message after to see if it's possible. But
Yeah, in fact, Avi put in the extending it where you could actually, I think, probably convert it yourself. That's my guess, knowing the way Avi built things, but he put it in the chat. But frankly, that's exactly what I was thinking. The playbook is a perfect place for this extension and this coordination. So I agree with that. So let's make sure we follow up on that, because I know that would be something very accessible. Should we do this with you guys?
Yeah, I think it'd be really useful if there were some sort of like, you know, ETL or translations made from the different the different cloud platforms, biomedical cloud platforms, so that you could collect all that data, right? So one of the things so we have a portal system at TAC, and so whenever you you run a job, right, you have data and you have your process, and then you even have like the hardware and that you're running it on. And there's a DOI that you can create that kind of collects all that together, you know, into a set of metadata about that. And I think that every cloud platform probably has something like that. And I think that's just the challenge is converting that into the CFDE playbook.
I agree. Other questions for our presenters? Okay, well, we put this on, and someone kindly noted actually that the last month's webinar for HubMap wasn't online. It now is. So ask and ye shall receive. We got that online. So thank you for asking about that. It was like the link wasn't working. So it's up there now. And this webinar will also be up on our Knowledge Center page, and we'll circulate that to everybody after this. So you can take a look if you want to comb back through this. And also, you know, if you didn't have a chance to do this, and also there'll be contacts in there for people if you want to follow up. And I think definitely this idea of the playbook is an awesome one. So Seth, we should communicate with Avi about that. And then Mackenzie, any final programming notes or things you want to wrap up on?
Just one last item. So thank you all for coming. Again, as Noelle said, this is recorded. Just want to give an update for our next webinar. Will be Friday, September 27th, and we'll have our presentation from xRNA. So we look forward to seeing you then, and please let me know if you have any questions or concerns in the meantime. So thank you, everyone.
Thanks. Happy long weekend. Thank you, Ari. Thank you, Martin. Thank you, Seth.
thank you guys.
Bye.